Thank you, and welcome to our webinar this morning on Translarna. I'm happy to say we have Craig McDonald from UC Davis on the call, Stuart Peltz, President and CEO of PTC Therapeutics, and Mark Bolding, who's Chief Executive Officer and Legal Counsel for PTC Therapeutics. We're going to talk about a lot of things this morning, to include the FDA and the process that is now being undertaken by PTC and the FDA. We're going to talk about the data and the trials and what happens next. So we're going to start with Craig McDonald, who's going to present the data on Adalind to date. Craig, over to you.
Great. Thank you, Pat. Good morning. My name is Craig McDonald. I'm a professor of pediatrics and rehabilitation medicine and a specialist in neuromuscular medicine at the University of California, Davis Medical Center. It's a pleasure to be with you today, and I look forward to presenting some of the emerging data in Duchenne muscular dystrophy natural history, as well as data from Adalind's clinical development, which shows clinically meaningful results with Adalind. Adalind actually targets a specific nonsense mutation or stop codon mutation. There's a very specific environment within the messenger RNA and the ribosome that Adalind works through. The medication specifically targets stop codon mutations. And it's important to point out there's really no other treatment available for this patient population in development. The Adalind promotes read-through of a stop codon mutation, and again, there's high specificity for nonsense read-through without affecting the normal termination codon or the normal stop signal within a gene. PTC really has an 18-year history with discovery and development of Adalind. This company was really the pioneer, being the first to perform a registration directed study. Like other ultra-rare drug development programs, PTC initiated their first large study, Study 007, really in the absence of natural history data and a limited number. There was a limited number of validated endpoints which were really available at the time. Subsequent to the completion of this first large-scale randomized control study, academic experts in the field and other companies have performed a number of studies which have increased our knowledge about the course of the disease and what is the best target population to evaluate a drug effect in a one-year study. I think it's important to point out that here the EMA approval occurred in July of 2014 for conditional approval, but the EMA draft DMD guidelines were not available until 2013, and the FDA draft guidelines, which PPMD was instrumental in getting the entire Duchenne community really valuable endpoint to develop those draft guidelines. Those were not available until 2014. The natural history publications really did not become available until about 2010. So when PTC embarked on this original study, Study 007, which was the first large registration trial conducted in 174 patients, they were essentially building the plane as they were flying it, so to speak, because a lot of natural history data and draft guidelines from the FDA and European Medicines Agency were not really available at this time. I want to just focus a bit on dystrophin production. With Adalind, the treatment actually results in a full-length functional dystrophin protein in animal models. This has been shown in the MDX animal model of Duchenne muscular dystrophy. And you can see here with the Adalind-treated MDX mice, there's significant productions of dystrophin. This has also been shown in the zebrafish model of Duchenne muscular dystrophy, untreated versus Adalind-treated zebrafish, and there's also been shown to be normalization in muscle protection. This is data on eccentric contraction-induced force decrements showing normalization with Adalind, and also a reduction in spilling of the serum creatine kinase into the periphery with Adalind-treated animals as well. So these results have been independently verified by a number of investigators, and I think it's important to point out that the animal models are showing functional changes in the muscle as well. Importantly, I think with regard to the original Phase 2A study, this was actually the study done in humans showing proof of concept with regard to Adalind's ability to produce dystrophin in human-treated patients. And PTC originally looked at dystrophin production in proof of concept to demonstrate that Adalind worked in their Phase 2A study. This was a study that actually treated patients for only 28 days, so only 4 weeks. You can see with many of the other exon skipping therapies that have been studied, such as Eteplirsen and Dystrophin, oftentimes sufficient levels of dystrophin are not produced until upwards of 12 to 24 weeks of treatment. But with Adalind, the proof of concept study actually showed that 61% of patients actually showed an increase in dystrophin staining, and with just 28 days of treatment with Adalind. So this Adalind really has a different mechanism of action. It's showing dystrophin levels earlier than perhaps we see with the other exon skipping strategies, and this may be one of the reasons why we've been able to see greater efficacy at 1 year with Adalind than we've seen with any of the other exon skipping strategies. And I'll get into that data in just a few minutes. In addition, PTC also looked at dystrophin and other proof of concept studies to demonstrate that it really worked even with a number of different stop codon mutations. You can see here that full-length dystrophin is produced in the nonsense mutation Duchenne dystrophy patients in patient myotubes grown in culture following Adalind treatment. You can see the increased levels of dystrophin produced in the myotubes grown in culture here, and this really has occurred no matter where the what the nature of the stop codon mutation is, with a variety of different stop codon mutations. So this is actually data from patients with different 6-minute walk tests with levels of dystrophin. These are just some examples of levels of dystrophin in 4 different patients at different ages. You can see that there really is very little correlation between the level of dystrophin and the actual distance of the 6-minute walk test. So there's not a straightforward degree of predictability between dystrophin expression and a clinical endpoint such as the 6-minute walk distance change. So I think that's important to point out just with regard to the limitation of dystrophin as a pharmacodynamic biomarker. It certainly does show very important proof of concept data that has been demonstrated with other exon skipping strategies such as with Eteplirsen. The next slide actually shows some very important data to consider, and that's really an experiment in nature. And this really addresses the question of how much dystrophin is really necessary to make a meaningful change to a patient. And what we're really learning is that very small increases in dystrophin above background can lead to meaningful improvements in disease progression with Duchenne patients. So there is an experiment of nature that actually consists of exon 44 skipping amenable patients. These patients actually have naturally occurring exon skipping that occurs without treatment that results in very low levels of dystrophin-positive revertant fibers. And I think it's important to point out that these patients actually behave differently than virtually any other Duchenne patients. And measures of disease progression, despite the fact that they have an out-of-frame mutation, measures of disease progression have shown these patients to be more mildly affected. So the data on the left actually shows Kaplan-Meier data on loss of ambulation from our Synergy Natural History Study, and the dark red survival curve actually shows the proportion of patients that continue walking with exon 44 skippable mutations who were actually steroid treated versus non-steroid treated versus all other mutations. What you can see is virtually 50% of all these patients treated on steroids are actually ambulating into the late teens and out to age 20 in our Synergy Natural History Study. And in fact, when you look at the North Star data, the North Star Ambulatory Assessment, which is another Duchenne-specific measure of disease progression, these patients actually have much less loss of function over 2 years in comparison to all other mutations. And so this really, these patients really do have very low levels of dystrophin, which I think is important to point out. Now, here's actually some Western blot data that has been provided courtesy of Judith Van Dudecom, who's a pioneering researcher in exon skipping. And these data are actually baseline biopsies in patients who are exon 44 amenable, who essentially have not been treated with any exon skipping strategy. And you can see that their Western blot values are less than 5% dystrophin levels. Both these patients, you can see the amount of dystrophin by Western blot. And despite these very low levels of dystrophin, these patients are continuing to do very well clinically. So this concept that you need 10% or 15% of dystrophin to have a meaningful change in clinical course, I think is really just not accurate, and perhaps levels even as low as 1 or 2% could really be very beneficial to a patient. If we go to the next slide here, this is actually data from a recently published manuscript that shows that there's with these exon 44 skipping patients, there's actually a very specific splicing factor that interacts with exon 45 that leads essentially to a conversion of a Duchenne genotype into a more Becker-like genotype. And so this really explains why these exon 44 amenable patients have really different natural histories. So, for example, they presented a patient who had an exon 45 deletion, which was out of frame. The patient was 18 years of age. They were walking independently with a waddling gait, showed some difficulty getting up the stairs and climbing stairs. Their North Star value, they were still ambulatory, was 8 out of 34, and their 6-minute walk distance was 252 meters at 18 years of age. So, again, this patient who was exon 44 amenable had very low levels of dystrophin by Western blot. In this case, it was about 7%. But again, doing very well in terms of clinical function. So with that, I'd like to shift and talk about natural history of Duchenne. Duchenne is manifested by a progressive loss of the ability to perform physical functions. These milestones are all interconnected with one another, and time of loss of function predicts time to the loss of the next function. So, for instance, the 4-stair climb actually will predict time to loss of the next function. And 4-stair climb actually predicts ultimately loss of ambulation, and loss of ambulation ultimately predicts time to ventilation. So the North Star data here, which is really a new Duchenne-specific measure of function, is a clinically meaningful function, which is predictive of future disease course, and was applied to study 020. This is the subsequent PTC Adalind trial, And this measure was applied shortly after the measure was validated. So I think it's important to point out that Duchenne is really a very complex, heterogeneous disease that requires multiple endpoints to understand a drug's benefit in a one-year study. You can't just focus on just one endpoint. You really have to look at a host of different endpoints to understand a drug's benefits. There's really no single endpoint that captures progression throughout the course of the disease. In addition, we need to understand which subset of Duchenne patients are best to apply very specific endpoints, such as the 6-minute walk test, over a one-year study, because we need to apply the specific endpoints to the very specific stages of the disease to be able to study a treatment effect over a one-year course of time. If we go to the next slide, this actually shows recent natural history data that was really only available in 2014 and demonstrates the challenges of the 6-minute walk test as a clinical endpoint in Duchenne dystrophy for 12-month trials, and show why really the mid-range of disease severity is the most appropriate population to study for a one-year trial. So this is actual 6-minute walk distance data published by Panay that actually demonstrated very little changes occurring in 6-minute walk distance if baseline values were above 400 meters. These patients actually changed very little over time. It wasn't really until you got out to 3 years that patients with baseline values of over 400 meters actually showed much decline. The right graph actually shows a comparison of 6-minute walk distance and MR spectroscopy data, actually, which has been presented by the Imaging D&D group. And really what you see here is that when the muscle fiber loss, which is indicated by an MR spectroscopy fat fraction, so as you get increased fat fraction, that is indicative of fiber loss. And when you get out to these critical threshold values of 80% or greater in fat fraction, which corresponds to a significant loss of muscle fibers, and you hit this critical threshold, and all of a sudden the 6-minute walk distances actually begin to plummet over time. And again, this occurs generally with a 6-minute walk distance less than 300 meters. So when you hit this critical threshold of 80% fat fraction, many of those patients have 6-minute walk distances below 300 meters, and you see this very heterogeneous but precipitous loss of 6-minute walk distance. On the other hand, patients who are between 400 and 300 meters actually show a much more predictable level of decline in terms of both 6-minute walk distance as well as their fat fraction values over time. There's also the natural history data I just showed you has also been confirmed in other studies. For instance, this is placebo data from the Eli Lilly Tadalville trial, which has recently been publicly presented by Ron Victor and his investigator team. This is data actually, just to remind you, the inclusion criteria for this study was 6-minute walk distances between 200 and 400 meters. And what you see here is when patients were below 300 meters, you get this very precipitous decline in many of the patients in 6-minute walk distance over time. Other patients are showing less of a decline over time. And so this data is really very consistent with the biologic plausibility showing that when you get below values of 300 meters, patients have less muscle substrate to treat in the lower extremities, and this is why some of these patients in the context of clinical trials are showing very precipitous drops and declines. And so this really demonstrates why the middle range, around 300 to 400 meters, is the most appropriate population for one-year studies using the 6-minute walk distance as an endpoint. I want to comment on what is a clinically meaningful change in 6-minute walk distance for Duchenne patients. A recent study that our group published, Eric Henriksen and others, really has demonstrated that when anchoring the 6-minute walk distance to health-related quality of life outcomes, This confirms that 6-minute walk distance changes of even 10 to 20 meters is clinically meaningful in terms of changes in PODCI, health-related quality of life scores, really across a wide range of ambulatory patients with Duchenne dystrophy. So again, changes of 6-minute walk distance over 1 year of 10 to 20 meters are clinically meaningful to Duchenne patients. The next slide really addresses the question of what is a clinically meaningful change in a time function test. Time function tests have been used for decades in Duchenne clinical trials, and this is recent data that we've gathered from the Synergy Natural History Study. This actually shows health-related quality of life scores versus 4-stair climb values really at 1.5-second increments. So you can see for every 1.5-second change in the 4-stair climb test, there are clinically meaningful reductions in quality of life that occur across the disease severity in Duchenne dystrophy. So even a 1.5-second change on a test such as the 4-stair climb, where the baseline value might be 5 seconds, a 1.5-second change, which might be 30 to 40% of a baseline value, is clinically meaningful to patients. If we could go to the next slide. This really shows what kind of changes we see with steroids in terms of time function tests, and really in the most contemporaneous clinical trial to date. This was data that was a study conducted in China recently and published in 2015, comparing prednisone-treated patients with placebo-treated patients. And in this study, over the course of a year's time, prednisone resulted in a 1.7-second change for 10-meter run-walk tests and a 1.6-second change for stair climbing. And so, again, we know the meaningful impact that steroids have had over the long term, over the course of the disease when you follow patients for many years. And in fact, what do these types of changes over 1 year, a 1.6 or 1.2 change, what do they translate to for a Duchenne patient longer term? And I think the Synergy Natural History data gives us some useful information here. And here in the Synergy Natural History data, when we look at patients with all mutations, a 1.6 to 1.7 second change over 12 months on a time function test such as the 4-stair climb really what does it translate to in terms of prolongation of ambulation? And this is the Synergy data comparing steroid-treated patients who've been treated for greater than a year on steroids. This is all mutation subtypes. Median age of loss of ambulation of 13.3 years versus those patients in the Synergy Natural History cohort who had less than 1 month of steroids, median loss of ambulation of 9.8 years. So really steroids have translated to a 3-year benefit in terms of prolongation of ambulation in this natural history trial. So you can see that these very subtle changes of 1.5 seconds, 1.6 seconds on a time function test can actually translate to substantial benefit for a Duchenne patient longer term. So again, just to summarize with regard to natural history, Duchenne is a complex heterogeneous disease requiring use of multiple endpoints in clinical studies of a 1-year duration. Both clinical endpoints and loss of milestones predict later disease progression, and then specifically the 6-minute walk test, the time function test, and the North Star actually will predict loss of ambulation. And 300 to 400 meter baseline values of the 6-minute walk distance endpoint is really the most appropriate range for a 1-year trial using the 6-minute walk distance as an endpoint. In addition, changes on the 6-minute walk distance of 10 to 20 meters are clinically meaningful across a wide range of ambulatory patients. And then finally, a change of 1 to 1.5 seconds in the time function tests and also functions lost on the North Star, which I'll get into here momentarily, really correspond to clinically meaningful changes to Duchenne patients. Now I'd like to get into the specific results of the Adalind trials. I think it's important to understand why study 020 failed, and really the largest reason was the failure to enrich for declining patients.
So, Craig, can you
yep.
just explain? I think it would be helpful for the group if you'd explain enrichment.
Yeah, so enrichment really pertains to, in the context of a clinical trial. If you're only going to do a clinical trial for a short period of time, say 12 months, enrichment would involve the inclusion of patients that over a 12-month period of time you would be most likely to show a drug effect in that shorter duration time. That doesn't mean that patients beyond that enrichment group aren't benefiting from the drug. It's just here, for instance, if you include stable patients above 400 meters in your trial, both the placebo patients and the treated patients are not really going to be changing much over the course of 12 months. Whereas if you include patients who are rapidly declining, some of those patients who are actually on a treatment, they may not produce enough dystrophin over a rapid enough period of time to really save the patient from losing ambulation, because you get these large precipitous declines that occur very abruptly. So by enrichment, we're really talking about this middle group here for the context of a 12-month clinical trial, which are showing more predictable levels of decline over the course of 12 months. And in fact, enrichment doesn't have to be the patients who get to participate in a clinical trial. Enrichment can be your pre-specified statistical analysis plan that actually demonstrates what patient population or which subgroup, say 300 to 400 meters, are you most likely going to be able to show a statistically significant drug effect over the course of a trial. And this is the actual placebo data from PTC's study 07-020. You can see very predictably in that 300 to 400 meter specified group, about 8 to 9% of patients lost ambulation. None of the patients lost ambulation, placebo patients above 400. When you get below 300 meters, in study 07, 31% lost ambulation. In study 020, 48% of the placebo group patient population lost ambulation. So in fact, the baseline, in terms of enrichment, this baseline 300 to 400 meter subgroup is the most consistent and reliable range to evaluate a drug effect in a one-year trial. Does that answer the question, Pat?
Yeah, I think yes, that's very helpful, Craig. One of the other questions around this is Becker patients that were included in the study, and perhaps we can get into that a bit later.
Yeah, and in fact, there were inclusion criteria. There were some Becker patients, just a couple that were included in study 007. In study 020, there were inclusion criteria that tried to eliminate the possibility of Becker patients being included. But I'll get into that here with this next slide.
Great, thank you.
And so it's important to understand here in this slide is why the study 007 actually failed to adequately enrich for declining patients here. And this was the study 007. So the mean 6-minute walk distance was about 356 meters. They actually took all comers, so 75 meters all the way up to 533 meters. And based on the data, there was actually a 31.3 meter treatment effect with Adalind in the ITT population. So the placebo patients declined 12 meters. The treated patients declined 44 meters. And they actually came up with inclusion criteria that were addressed to try to eliminate the possibility of Becker patients being included. They actually increased the distance walked to 150 meters. And they used an inclusion criteria of 80% predicted. And then applying those data to the 007 study population, there was actually a nearly 50-meter treatment effect, a statistically significant p-value with Adalind. And these are the criteria that were actually used to develop the study criteria for study 020 that PTC subsequently conducted. Now, if we go to the next slide, this shows actually the fact that the inclusion criteria of the used for study 020, which was this greater than 7 years, 150 meters, and less than 80% predicted, unfortunately didn't adequately enrich for patients who were in the declining phase of the disease. And in fact, whereas the original ADP population from study 07, if you look at that population, their 6-minute walk distance was 340 meters. The actual study 020 population actually went from 143 meters all the way up to 526 meters. And in fact, the baseline value was 364 meters. And so in fact, the ITT population actually didn't really look like the ADP population at all. It looked really more similar, in fact, to the ITT population of 007. And in fact, when they looked at the entire population then in study 020, because of this failure to enrich for declining patients, the patients had values clear out to 526 meters, and many patients were included who had higher 6-minute walk distances. The delta value that was obtained was only about a treatment effect of 15 meters, which did not reach statistical significance. Now, previously or subsequently, actually, a nonlinear model has been applied, which takes into account this precipitous decline in function for those below 300 meters. And in fact, by applying that nonlinear model statistically, that gave a 14-meter treatment effect with a statistically significant p-value of 0.04. But this was a post-hoc analysis. This was not a pre-specified statistical analysis.
And in general, Craig, the FDA does not appreciate or accept the post-hoc analysis. Is that
yeah,
the case?
Exactly. The FDA has been very clear that they really will really only accept pre-specified analyses that are pre-specified in a statistical analysis plan before the data is unblinded. They will basically say that post-hoc types of analyses are important and can be used for hypothesis generation, but not to definitively demonstrate a drug effect. And I think that's been a very firm position from the FDA applied to many studies.
And so, Craig, could you make one comment on 80% predicted and how that's applied?
Yeah, so going back to these original 80% predicted criteria, this was actually, the reason this was applied in study 020 was there was a goal to actually eliminate patients with Becker muscular dystrophy. And in general, in the pediatric age ranges, essentially all Becker patients have 6-minute walk distances above 80% predicted value, which is sort of a age-related percent predicted value, similar to a percent predicted forced vital capacity on a pulmonary function test. And Becker patients will generally be above 80%. So PTC actually adopted that as a criteria. In addition, they didn't want to adopt an upper limit of 6-minute walk distance of, say, 425 meters or 450 meters because there was a concern that perhaps patients, in order to get into the trial, there might be just a natural tendency. We can all appreciate this, a natural tendency if somebody was really high functioning to perhaps maybe not give the best effort possible in order to get into the trial, whereas the 80% predicted value really might eliminate that as a potential bias issue that could affect patients' levels of effort and so forth. But in fact, obviously the 80% predicted value didn't work because it really, because of different heights and different limb lengths and so forth, there were patients who were actually below 80% who had very high values of 6-minute walk distance of greater than 500 meters. So in fact, the ADP criteria, the 80% criteria, just didn't work out. It wasn't a successful inclusion criteria for the PTC trial. That's not to say anything about the effectiveness of the drug and whether the drug actually works versus placebo. So again, this is more of a summary slide. So essentially, with the 3 studies, the 007 study, which had the entire population, went from 75 meters to 533 meters, mean 6-minute walk distance of 356 meters. The study 007's ADP criteria, which is a decline phase criteria, didn't adequately enrich. And then finally, the study 020 ITT population here with a 6-minute walk distance of 364 meters. So really, we had rapidly progressing patients that can decline over just a matter of weeks in spite treatment. And there's also stable patients. And so in terms of a pre-specified statistical analysis plan, what really makes most sense is to try to get consistency between the studies and focus on patients who have 6-minute walk distances which are more predictable between 300 and 400 meters, which actually includes over almost half of the entire study population. So this is not a small subgroup. It's actually virtually half of the entire study subgroup. So this baseline 300 to 400 meter subgroup is that group that, at least in a one-year study, gives you the most consistent and reliable changes over time in order to evaluate a drug effect. If we go to the next study, this is actually a study from 020, which actually demonstrates that if you look at that patient population who had a 300 to 400 meter subgroup, they actually demonstrated a 47-meter benefit with Adalind, which was statistically significant. And if you adjust for multiple comparisons using what are called statistical permutation methods, which is a more rigorous methodology to get a p-value, a statistically significant p-value, even with that, the p-value is still 0.036. Again, demonstrating a significant Adalind treatment effect of 47 meters over a one-year study time in that 300 to 400 meter subgroup. And again, this is not too distinct from the criteria that are being used right now for the Teplerson confirmatory study. That population in that study actually ranges from 300 meters to 450 meters over time. So these criteria are actually not too different than what's being used in current studies. If we go to the next slide, if we then look at whether the 2 studies that PTC conducted showed consistent results in comparable populations, this actually shows the values obtained in study 007 in the 300 to 400 meter subgroup, and it showed a 51-meter treatment effect with a p-value of 0.026 in study 007, and in fact almost identical results in study 020, a larger population of 99 patients. We get a treatment effect of 47 meters with a p-value of 0.007. So again, looking at comparable populations between the 2 studies, we actually we see actually very significant treatment effects. And I should emphasize that the 300 to 400 meter subgroup was actually pre-specified by PTC prior to the unblinding of that study. It was actually pre-specified in their statistical analysis plan. So this is not a post hoc analysis. This is actually pre-specified analysis that was part of the original statistical analysis plan put forth by PTC. If we go to the next study, this actually shows the time function test results, really showing very consistent results between the 2 studies, 007 in the blue and 020 in the orange. And what you see here is particularly in the 4-stair climb and 4-stair descent, we see significant and meaningful effects really across both studies with Adalind treatment. And these benefits, even though it sounds like only a small number of seconds, they actually pertain to anywhere from 29% up to 38% of the baseline time spent performing those tests. So we're actually seeing treatment effects on the order of 29% to 38% on these time function tests. We see effects of about 18% to 21% in the 10-meter walk-run test. So we're seeing consistent results really across all of the time function tests in the ITT population, which was all the patients studied with Adalind treatment. Now, if we look at these time function tests with that 300 to 400 meter subgroup, we see even more substantial changes. And here, the changes on the treatment effect, the treatment benefit was on the order of 64%, 63-64% of baseline values with changes greater than 3 seconds with a 4-stair climb, all the way up to 87% or 100% effects versus baseline values with changes on the order of 4 seconds, all statistically significant. And then even in the 10-meter walk-run test, we see changes of 32% to 51%. Again, looking at that pre-specified subgroup of 300 to 400 meters with the time function test. So this is really the first study that's ever been conducted in Duchenne with a dystrophin restoration strategy that has shown consistent benefits across all of the time function tests in a 12-month study. If we go to a meta-analysis, which is a way to really combine the data from study 007 and study 020 and see what kinds of effects do we see across all of the patients studied. And again, there was a meta-analysis which was actually pre-specified as part of the PTC statistical analysis plan. But looking at the meta-analysis, we see about a 1.3-second change, statistically significant with 10-meter walk-run, a 1.8-second change with stair climb, and a 1.9-second change, statistically significant with stair descent, and about a 20-meter treatment benefit, again, statistically significant with the 6-minute walk test using, again, a meta-analysis, which was actually pre-specified as part of the statistical analysis plan. So again, with the PTC data, if you look at the totality of the data, really shows clinical benefit across the primary endpoint, which is the 6-minute walk distance. So this is the meta-analysis, study 020 and the study 007. And then with the across all of the time function tests, you also see very consistent treatment benefits favoring Adalind versus placebo, and these are 95% confidence intervals. You can see that on very few of these measures does that 95% confidence interval reach 0 or below. The results are very consistent and all favor improvement in disease progression with Adalind treatment. Now, I'd like to shift focus to an emerging endpoint that's been used in Duchenne trials, and that's the North Star Ambulatory Assessment. And this is a composite endpoint which evaluates physical function across 17 tests with increasing difficulty for a Duchenne patient. So if you start for Duchenne patients, the most difficult functions to perform, and those that are lost earliest, are functions such as hopping on one leg, rising from the floor, running, jumping. The less difficult functions, which are lost later in the disease course, are things like getting up from the chair, walking, and standing. And essentially, with what we've done here with this data set, and I think the the academic experts in the Duchenne community are really getting excited about this approach to North Star data, is looking at the North Star data in terms of each of these important functions being essentially an important, clinically meaningful function that is lost during the course of the disease. So you can actually think about it as a milestone that's lost. And with the North Star scoring, you actually score each of these items from 2, able to perform normally, to 1, performs with difficulty, to a 0, unable to perform, where you have complete loss of function. So essentially what PTC myself, Eugenia McCurry, were actually providing some input on this. Well, essentially what they did is a shift analysis looking at the percentages of patients who actually started with baseline values of either a 1 or a 2 who actually shifted and lost that function over the course of the trial. And if we actually look at the data in that manner, what's really remarkable is that in 16 out of 17 of the functions, fewer Adalind-treated patients lost those functions in comparison to placebo-treated patients. So this is each function. The orange bars are the percentages of patients who lost those functions treated with Adalind versus the gray bars are the percentages of patients that lost these functions who were placebo-treated patients. And in fact, when you look at this, the only one that actually didn't favor Adalind was lifting head, and that has actually been shown to not really relate to the others by what's called a rash analysis, which is, is it a valid measure of disease progression that in the context of all these levels of function. But basically, 16 out of 17 of the functions, fewer Adalind patients actually lost these functions. And in fact, there was a 31% reduction in loss of function with patients treated with Adalind across the entire ITT population, not just the 300 to 400 meter subgroup, but really all the patients with Duchenne dystrophy with a statistically significant p-value of 0.01. So where the North Star was actually was a pre-specified value, there was about a 1-point change on the overall North Star, which just averages all this data favoring Adalind. But when you look at the 300 to 400 meter subgroup, there was, which was the pre-specified subgroup, there was about a 4.5-point change score on the North Star. But we're realizing that actually looking at the North Star data in this fashion actually is a much more meaningful way to look at the data. We lose a lot of granularity by just averaging all these values across time. And in fact, we get a better p-value and a 31% reduction in risk of loss of function by analyzing the North Star data in this manner.
Craig,
what about?
Craig, one question here is, could you just explain to the group where the North Star was, when and in what studies the North Star was used, and was it exploratory secondary endpoint?
Yes, that's a great question. The North Star was actually developed and validated by a European group of physiotherapists, and actually was not available in study 007. It was only used as one of the secondary endpoints for study 020, and it's really just until really this year that we've come to realize that perhaps different methodology for evaluating this North Star data is actually perhaps more meaningful and more provides more granularity in terms of actual loss of function and clinical meaningfulness and using it as a measure of disease progression. But when the study 020 was started, really what we had at that point in time was really just the total North Star score, which was just a sum of the zeros, ones, and two values across the 17 functions. Moving to again loss of clinically meaningful functions, there's one function which actually isn't included in the North Star, and that's the ability to climb stairs and descend stairs. And in fact, if you look at the study 020 in the ITT population, this is across the entire population. This is the percentages of patients who actually lost four-stair climb. And you can see that about 8.9% more placebo patients lose stair climbing ability, and about 7.9% more placebo patients lose stair descending ability over the course of the trial, which was nearly statistically significant. So this data is actually very consistent with what we're seeing in the North Star data as well. And in fact, if you look at the patients who actually lose the ability to ambulate altogether, this is data from the less than 300-meter baseline subgroup. So you can see that there's trends favoring Adalind, but many of the patients in both groups are losing ambulation. But what's remarkable is if you look at that 300, we know that no patients above 400 meters lost ambulation in either the placebo group or the Adalind group. But when we look at the 300 to 400 meter baseline group, no Adalind patients lost ambulation, whereas 9% of placebo patients in study 007 lost ambulation, and 8% of placebo-treated patients in study 020 lost ambulation. So I think it's really remarkable that really across these two studies, Adalind is really protecting against loss of ambulation in that 300 to 400 meter subgroup. And we know that patients above 400 meters will eventually progress to that 300 to 400 meter window. And so I think this really has meaningfulness across the spectrum of disease with Duchenne muscular dystrophy. So again, to summarize with regard to safety, I think Adalind has really a remarkable safety profile. Over 400 Duchenne patients treated in randomized studies. The most commonly reported adverse reactions in these studies were just mild increases in vomiting, diarrhea, nausea, headache, upper abdominal pain, and gas. Fewer than 1% of patients discontinued Adalind due to adverse events. And again, with the commercial experience in Europe with Adalind, the compliance has exceeded 90% of patients on commercial drug in Europe. So over 600 Duchenne patients treated. And now there's been over 500 cystic fibrosis patients who have also been treated with Adalind. 2 large placebo-controlled phase 3 studies. One hasn't been, the results aren't available yet. But again, there's substantial experience in over 1,000 patients dating back to 2009 showing that this drug is really very, very safe. So really, to me, the benefit-risk considerations are really quite straightforward. There definitely are benefits to the disease, and there really are no risks. And so I'd like to focus a little bit on just this whole issue of benefit-risk here. If we look at, for instance, Adalind, it demonstrates clinically meaningful improvements in disease progression, preservation of function, difficulty in ambulation, 6-minute walk distance. There's, in the 300 to 400 meter subgroup, about a 47 to 51 meter treatment effect. In the meta-analysis, about a 20 meter treatment effect. Impaired in terms of functions assessed by the North Star, there's a 31% reduction in risk of losing functions based on the. If we look at both stair climbing and stair descending, there's a 1.8 to 2.4 second change in the ITT population, both in 4-stair climb and 4-stair descend. We know that's a clinically meaningful value. Even if we look at both the 300 to 400 meter population and the less than 300 meter population with a 10 meter walk run, there are benefits in both those subgroups favoring Adalind. The 10 meter walk run is really an indicator of minimal ambulation, which is lost subsequent to the 6-minute walk distance. So if you look at even those more severely impaired Duchenne patients who are less than 300 meters, they're still showing a trend on the order of 1.2 to 2.6 seconds favoring Adalind with the 10-meter walk-run test. And then in terms of loss of ambulation, in the 300 to 400 meter subgroup, no patients losing ambulation. And then finally, this is data that I didn't present, but PTC now has some extension phase data which actually demonstrates in the more severely affected Adalind-treated patients stabilization of pulmonary function with a reduction in loss of absolute forced vital capacity by about 13.8% versus the Synergy natural history data set. So I think it's clear that really across multiple endpoints, Adalind is showing meaningful changes. Just to remind you, with the recent randomized placebo-controlled trial with prednisone, this is a more modern study but again, a treatment effect on the order of 1.6 to 1.7 seconds on time function tests. We're seeing this magnitude of effect of Adalind on top of steroids in study 020. And so that's, I think, very encouraging. And just to remind you that this level of magnitude improvement in a time function test, at least with the Synergy natural history data, actually translates to about a 3-year prolongation in ambulation, which we know ultimately is predictive of later time to loss of upper limb function and later time to onset of ventilation. And then finally, this is just data to just food for thought here, but we know the encouraging results that we see with eteplirsen with longer-term study of patients on out to 3 to 4 years, and also with drisapersen, we know that with longer-term administration, we've seen effectiveness. There were obviously safety concerns. But if you just look at the 12-month treatment effects across the multiple primary and secondary endpoints with dystrophin restoration, the green results actually demonstrate a treatment effect favoring the dystrophin restoration drug. The red actually shows results favoring placebo over historical controls. And this is not to say that I actually testified on behalf of both drisapersen and eteplirsen at the 2 AdCom committees. So I believe these drugs work and are a benefit to Duchenne patients. But in the context of a 12-month study, just to remind people, with drisapersen, with their pivotal phase 3 trial, they got about a 10-meter treatment effect. There was actually no treatment effect on stair climb or on 10-meter walk run, and a very small change in stair descend. Eteplirsen versus the Italian and Belgium cohorts historical control data at 1 year actually showed that there was no treatment effect of eteplirsen. We all know that on longer term follow-up, 3 to 4 years, there seems to be a robust treatment effect. And with, again, the North Star, there was about a 1.6 second decline. With Adalind, really across all of the time function tests and across the 6-minute walk test, there is a meaningful and significant benefit favoring Adalind-treated patients. And again, we know that even at 28 days, a substantial percentage of the patients are already showing dystrophin production based on their phase 2A proof of concept study. Now, it may be low level of dystrophin, but we know low levels can really create meaningful benefit. And here you see it at 12 months across multiple time function tests, as well as the 6-minute walk distance. So again, I think with exon skipping drugs such as eteplirsen or drisapersen, we have to do longer term studies of perhaps longer duration, greater than a year, to demonstrate that treatment effect. But with Adalind, it's really been the first and only dystrophin restoration strategy that's shown the consistent benefits at a 12-month time point. And so with that, I will conclude, and I believe Stu Peltz might be on the line to make some additional comments.
Yeah, thank you, Craig. That was really excellent and a very nice review and comparison with the other studies that have been done on dystrophin restoration studies. So I know Stu and Mark are on the call as well. So I'm going to just start with a couple basic questions that have been confusing to the community. In the early study, in the first study, I think it was 007. The EDB muscle, the extensor digitorum brevis muscle, a small muscle in the foot, was removed for dystrophin for biopsy. In the later studies, the biopsy sites were basically in the quadriceps muscle and so first of all, could Mark or Stu or you, Craig, answer the question why a whole muscle and why was there such difficulty with the other studies in terms of biopsies to the point that at a certain moment you did not do any further biopsies?
Yeah, hi. Thanks for that question, Pat, and I think it's a very good one. And you have to remember, we were the pioneers where we started this where, as Craig has shown in a slide, and where we discovered Adalind for 1998 through 2003 or 4, and then we did a proof of concept study in a phase 2A study, just like other companies have you seen, where it's a small number of patients that we did it and very few highly selected sites who are experts in the arena. So the patients go there. We thought hard about what muscle initially to do, and we took, like you said, the extensor digitorum brevis muscle, and this was the phase 2A study. It was actually study 004 that this was done on, and we thought about the reason we did this because it's a small and vestigial muscle, so it's not really required just above the upper toe, where you can actually remove the small little muscle and therefore be able to interrogate the whole muscle as a consequence of that. It's not highly involved within the disease state, so you can get we thought a reasonable idea of the dystrophin amount of protein in that. So this was in 2004 and 5 or so, where we were doing this, and we were able to look at the whole muscle, and it really was done just in a few sites, and it was all done really carefully, and we were able to do that, and that's where we were able to show quite nicely that we demonstrated dystrophin production within that. And you can see, actually, Craig showed a nice set of slides. I think he showed about 6 examples where not only could we do that, we took some of those, not only do you see it in the treatment in the patients, but if you actually grow the myotubes from patients in culture and actually ask, do you see dystrophin production occur when you add Adalind, the answer was yes. And I think what was important is we saw, as Craig had showed, in 61% of the patients where we saw that. But actually in myotubes, I think it was we did 35 cultured myotubes, and in all of those that we tested, we demonstrated that dystrophin production occurred. And you can see what was really quite nice in the slide that Craig showed. It didn't matter what exon the nonsense mutation was. It didn't matter which type of mutation it was. It was all 3 of the types of nonsense mutations. So it showed that really Adalind was able to produce dystrophin in all the myotube cultures that we did. So that we did that, and we had multiple discussions with both regulators, the European and U.S. regulators, and it was very clear to us in those discussions, while dystrophin is a very important proof of concept study, it is not at least we were told that it was not for approvals, and that we were required to actually define a clinical endpoint and use that clinical endpoint. And by the way, they told us none of the clinical endpoints at the time of what people were using would be satisfactory, so go define one. And that's where we, with the community, and in particular Craig McDonald, where we went and did the 6-minute walk test, showed that it was test and retest was good and reliable, and that's what we used as the primary endpoint with multiple other endpoints in order to the study it in 007. So that was the first study. And there we did say exploratory, and we knew dystrophin would be hard because we had so many sites. There were over 30, probably around 37 sites, and we knew that was technically challenging because the protein is unstable. You're having now a fair number of surgeons to be able to take different parts of the muscles. You have to ship it multiple times because they're where they are. And then you couldn't use the EDB muscle anymore, the extension digitorum brevis muscle, because your primary endpoint is 6-minute walk test. So you didn't want to actually do any biopsies on the kid's feet because we thought that could actually affect the primary outcome. And then you start using the quadriceps and other muscles, the quadriceps, and there we know it's highly variable. You're going into different arms with different variability in the muscles. So it's a highly variable test, and it's labeled. And with all those problems, there were huge technical problems that made it difficult to analyze. And this turns out to be the case. You could do it. It's very clear you could do some of these types of studies in single site settings, and you could get some results. But it's very difficult to be able to do this in a large phase 3 study. We've seen this as a problem. The BioMarin has seen it. Actually, there's multiple people who have done this now who it's been very clear that this type of biomarker is not amenable to be able to do it in a large phase 3 setting. And that's what, was the learnings from that. So I think that's sort of the history of this as a biomarker.
Thank you. So, Stu, you and Craig, you also did the MRI study alongside of this I think it was the phase 2B trial. So did the MR study results correlate with what you're seeing on functional outcomes?
Yeah, that was I think a side study by University of Florida that was measuring. So I think it really wasn't part of the 007 study in that sense. They were looking at a bunch of kids. I think, Craig, you may want to comment a little bit on that.
Yeah, I think that certainly I think the MR is a emerging biomarker that I think has been really useful. We're learning a lot from it. I think that most of the studies have used perhaps parallel designs where a proportion of the population are studied using the MRI biomarker. But if you're doing a worldwide clinical trial in 50 sites or in multiple nations, it's substantial difficulty, I think, trying to apply that biomarker, at least at this day and age. I think for smaller directed studies, I think particularly if you're looking at mechanisms of action where the medicated, the therapeutic may increase muscle mass or decrease inflammation, I think the MR biomarker, I think, is really quite useful. I don't think there's any data that shows that small changes in dystrophin on the order of what we see with a dystrophin restoration therapeutic can actually necessarily be picked up by an MR biomarker. And so I think that the jury's really still out on that. But I think MR imaging, and a lot of this work's been done by the Imaging DMD group with Krista Vandenborn and Lee Sweeney and their colleagues. This is really a very exciting technology and very exciting biomarker that's emerging that I think we'll see applied to increasingly in future trials.
Great, thank you. So let's shift a little bit of direction to the trials themselves that are people are in the extension trials now for many, many years, and there is great concern because you do have a approval in Europe and not an approval in the U.S. And we also are well aware that you've started a study or you're initiating a study in very young 2 to 5-year-olds. So one of the questions is, will the FDA want to see those results before giving you a decision?
Yeah, sure. Yeah, thank you. That's a good question. And I should remind everyone, we've been actually doing extension studies after trials. Clearly, kids almost up to 9 years. So we've been doing that and have held, for that for quite some time. In Europe, obviously, we have an approval, so to patients, it's commercially available to those patients. And those who are on section studies, we've been moving towards commercial drug. In case of the 2 to 5-year-old, the purpose there is part of the commitment process that we had with the Europeans to go into pediatric patients even lower than 5 years of age, which we're doing. It'll hopefully then allow the expansion of the commercial label and license to patients to greater than to those that even go down to 2 years of age. So we're encouraged by that as well. And I think when you think about it from the big picture point of view, that's mainly a PK and safety study. So that's the importance of that study. In part, what the regulators would like to know is when you expose kids younger than 5 years of age, is the drug different in terms of the levels within the blood that would be with those kids older than 5. And that's the main purpose of that trial. It's not an efficacy trial. It's really what they call a pharmacokinetic exposure and safety trial. So that's the purpose of that trial. In the case of the U.S., obviously, I think everyone knows we have had a refusal to file, and as we've announced recently that we're appealing that decision. We've formally done that, and we're hopeful that it's very important to be able to have discussions with the FDA to be able to try and reverse that decision, and that's what we're in right now.
Right. Thanks, Stu, for that. Can you talk a little bit about the process? So you've issued an appeal, and that then changes what happens next. So could you talk to us about what happens next now that that appeal is in process, the time it takes for this, and when will we know something?
So I'll tell you, let me ask Mark just to talk a little bit about the process. He's been spearheading this process.
Yes, hey everyone, this is Mark Golding, and I'm the Chief Legal Officer and also EVP, and regulatory reports up to me. So we've been, working for quite a while on the different choices here. There were a number of ways that we could have addressed this unfortunate RTF letter from FDA. But really, at the end of the day, we've gone through the appeals process. The formal name for it is the formal dispute resolution process. That might sound a bit scary, but it really actually is the process that FDA itself has created for exactly this situation, and they've actually publicly commented a couple times that they'd like you, this is something you should think about doing when you have an RTF that you disagree with. And so it's a defined structure, and what it really does is it escalates the discussion about our filing up to the next level of FDA management. So the decision was at the Division of Neurology Products level, I think that those of you that looked at the briefing books from the Dystrophin Adcoms kind of have a sense of how that went. And this escalates it up to the next level of center management, and we're hopeful that that will enable us to have a broader conversation about exactly some of the data that Craig presented and that we're aware of as well, to make our case that this application deserves a full review by FDA.
So obviously as I imagine you do as well, think it was unfair and wrong to give us the refusal to file letter, and that's why we're appealing it. And we're doing so we really believe in this. So it's very important for us to push it through this process.
Thanks for all that. So what in our community, we'd like to do something to help you. What would that be, and how would you see that happening?
Yeah. So I think, thanks for that. I think in rare diseases like, and ultra-orphan diseases like Duchenne muscular dystrophy, and even when you think about it, we're taking a slice of the Duchenne population, it's only 13%, that we really have to get the FDA to look at the totality of the data that includes not only just the primary endpoint, but the totality. You could see, I thought Craig did a very nice job in putting together, showing on the clinical data in a one-year study. No other study has shown both an improvement in 6-minute walk test, time function test, North Star, the activities of daily living. You go down the list, everything was positive. And when you look at the totality of that data, it seems to us that it's very clear that this drug is helping these kids. And that, along with what we'll be presenting later in terms of other data, we think is a very consistent story that actually demonstrates Adalind's activity. But I also think, and with Fadasia, it's very important for them to also hear what Adalind is doing for your children who have Duchenne muscular dystrophy due to a nonsense mutation. So I think that's very important for them to do, and I think it's very important for them to know this in the time frame. And time matters here because we're in the process. It's not that long of a process, and I think, it's always been our goal to make sure kids were on drugs, but I think now's the time to get this community organized to be able to say, if you see benefits to the patients on Adalind, and many of you have been on for many, many years, to actually communicate that to the FDA so that they know that that a removal of this or not being able to get the drug would be very bad for your children. And I think that's very important for them to get that message. And I think one thing you could do is, if you want, I'm sure, Pat, you're involved with that. You can talk to Mary Frances Harmon here at PTC, who has the advocacy group. But I think it's very important, again, for them to get this message and to get it now.
Thanks, Stu. And also keep in mind that on our website, we have Ryan has posted a survey to understand your experience if you've had one on Adalind Translarna, so that we can understand and share with the FDA what the experiences have been in the long term, because all of the individuals in the study have been on this extension, some of them 8 to 10 years, and none of them, or very few of them, have dropped out of the study. So I think that loyalty to this drug says something in itself. And I also know that it is true, as we've seen in eteplirsen and driftversine and Translarna, that we know in the long haul, even with steroids, that it takes a longer period of time for us to see the full impact. So, and these patients have been on this for years. And so one of the questions, Stu, is in this process of appeal and discussions, we don't, obviously there's no time frame for it in terms of the appeal happens and this happens and we'll get an answer, because I understand there could be several appeals going on. So the patients are naturally worried about you discontinuing the drug in the United States, which seems incredibly unfair and frustrating that's approved in Europe and not in the United States. So can you shed a little light on your plans and your timeline so that we have some understanding?
Sure. So let me, I think what you said is very important, and frankly, with the Fidasia and what they're saying in terms of that, what patients think of the drug are very important, and to have you collect that data is incredibly important. So anyone on the call who's on the drug, it'd be very important for them to fill that out as an important aspect to help us to show to the FDA what it's doing for their children. As you know, and as you pointed out, over the last 10 years or so, we've been using extension studies to supply children, the boys and young men with DMD, Translarna, right? And that we at this time haven't changed that, but there will come a time when if we're not able to reverse that simply because of resources and what's available to us, we'll have to reconsider how to do this and what we can do. We can't, in the absence of being able to do this, we simply don't have the resources in the long run to be able to continue forever. So now's the time, and I'm telling you this, really to take action and, in a sense, to work to make sure this doesn't happen and help us let the FDA know what it's doing for your children.
Great, thank you. So is there a timeline, a beginning and an end to the process of appeal, and what happens, and I know there can be another appeal, but is there a specific timeline that you have either on PTC side or on the FDA side?
So so point number one, I think it's the time, if you want to do something, the time is now. It's in the next few weeks is what I would do something. But I'll let Mark talk more about, in general, the timeline.
Yeah, sure. So there's a guidance document that sets the parameters and timeframes for this. It's, it's available publicly. It's a draft, as are many FDA guidances, and it gives a rough sense. And this is a process that takes, if you kind of follow it normally, a month or two. But of course, that's per cycle, and there could be more than one cycle. But I think actually more important than the specific days is FDA does have a commitment to trying to resolve these, fairly quickly, and that just accentuates Stuart's request that, if you have things that you think would be helpful, please, share them with Pat or share them with Mary Frances, because now is the moment to get that in. And I think it's always easier. The more cycles you go through, the more times you are on the other side of something from somebody, the harder they get dug in. So the first cycle, I think, is your best window for changing minds. So that's just another reason to get those stories in and help us present information to the FDA and to FDA management in particular that can help them come to a conclusion to reverse this decision.
Right. Thanks, Mark. And Mark, so the appeal process is basically to get on their agenda for review, which would then fall into the review process, AdCom, and decision. Is that what you're?
Yes, we're asking. Yeah, to be clear, we're simply asking for what two other companies who recently filed applications with similar challenges, frankly, in the data sets got. We want to be taken to full review. We think it's only in the context of a full review that you can appreciate some of the points that Craig highlighted in his presentation and come to the right conclusion on risk benefit. Really, an RTF is not supposed to include a substantive review at all. It's really supposed to be about facial completeness of the application. So we think that involving the FDA in a full review, having an advisory committee meeting, bringing in outside experts like Craig and others, bringing in clinicians with experience, bringing in the patient community with their own experiences and stories, all that is tremendously important for the FDA to make an informed decision about whether or not to ultimately approve the drug. We're just asking them to open the book and read it.
Right. Well, thank you, Mark. I totally agree with that. I think rare disease is really different, and they haven't seen many rare disease indications come through their desk. And I think it's really important for them to be able to be open and read books. So just a couple more questions. I know we're going way over. But this is really about Adalind, and this is probably more for Stu or Craig. Will Adalind help with a nonsense mutation caused by a frame shift?
Yeah. Thank you. That's a good question. And the answer is no. A frame shift leads to, in a sense, the frame, when you talk about frame, it's really talking about, in a sense, what amino acids make up the protein. And when you're in the right frame, you make the dystrophin. When you're in the wrong frame, you don't make dystrophin. It's like making a gobbledygook protein. So even if you read through the gobbledygook that hits the nonsense mutation, it still doesn't make the right protein and won't be beneficial. So you need to have a specific nonsense mutation, which is usually a single point alteration that leads to a nonsense mutation in the DNA that causes a premature stop. But if you then read through that premature stop, it goes on and makes the right protein, and that's why you see the dystrophin being produced. And it's important to point out when Craig shows you those dystrophin proteins that are being made, it's using an antibody that detects the end of the protein. It's called the carboxyl terminal end. So we know it's making the full-length protein correctly. So that's why we're sure that it's promoting what we call read-through or at the nonsense mutation and then goes on and makes the dystrophin protein.
Right. Thank you. So last question, because we're now about a half an hour over, is have you collected any quality of life data across those extension studies?
Yes. So yes, we have done the quality of life data activities of daily living, where we've all seen, and I think we even have posters that we've done on these that demonstrate that patients did more favorable on Adalind versus placebo. So they felt better. So we've had quality of life as well. We've done it in all studies, both 007, 020, as well as within other studies as well. And so we have that as well.
Great, thank you. I think there are no further questions, and this has really been a very comprehensive. So I thank all of you, Craig and Stu and Mark, for staying with us for the hour and a half and answering questions. Should more come in, we will send them to you, and we will report back through the website. But thank you very much, Stu. We're very enthusiastic about. We've come a long way with you from when we were in the plane flying it with really no understanding of how to do that. So I think what you've done and what you've learned over time has contributed to all the other companies has been very helpful. And certainly we've learned a lot about natural history and outcome measures from those first early days. So I thank you for that. I thank you for sticking through us and with us for those 10 years. And we certainly wish you success, and we're going to be very supportive to help the FDA understand the importance and relevance of Translarna for our community. So thank you once again, and this will be posted on our site within about 5 days. Thank you all.
Thank you.
Thank you.
Yeah, thanks.